Good morning. My name is Zaks, and I'm the Chief Medical Officer at Moderna. On behalf of myself and my colleagues, I'd like to thank the committee and FDA for the opportunity to present our data today. We've carefully watched and listened to the meeting last week, and in preparing our presentation for today, we've attempted to proactively address many of the topics raised at that meeting and in the days that have followed. We've been developing our mRNA 1273 vaccine with a goal to seek global licensure for the prevention of COVID-19 disease, and are here today seeking emergency use authorization based on phase 3 safety and efficacy data. I don't need to belabor the damage this virus continues to wreak directly on our health and indirectly on our society and our way of life. Since the pandemic began, we at Moderna have moved rapidly to leverage the advantages of our mRNA platform, and we've been working closely with colleagues from the National Institutes of Health to develop our vaccine. We've done so in a very transparent manner, sharing our phase 3 clinical trial protocol as well as recruitment metrics with the public. Let me briefly explain the merits of our vaccine. mRNA 1273 is based on messenger RNA, a molecule that is fundamental to the biology of every living cell and serves as the blueprint for all protein synthesis. Our vaccine uses our body's own cells to activate the immune system. It enables these cells to make only the part of the virus that is critical for the immune system to recognize, in this case, the spike protein. Importantly, our vaccine platform has some inherent safety features. The mRNA does not self-replicate, does not enter the nucleus, and does not integrate into our DNA. The manufacturing process is cell-free. It does not use products of animal or human origin, and it does not contain preservatives or adjuvants, thus avoiding some of the potential concerns of older vaccine technologies. Now, mRNA 1273 is not our first infectious disease vaccine. In fact, we've been in early phase clinical trials for the past 5 years, conducting 12 clinical trials that have enrolled over 1,700 healthy volunteers. SARS-CoV-2 is the 9th virus against which our mRNA vaccines have elicited neutralizing antibodies, and we have not seen a significant safety concern in any of our trials to date. Since the company's inception, we've been investing heavily in understanding the critical quality attributes of our mRNA medicine, and we have been using these insights to continuously improve our process and manufacturing capabilities. We've leveraged this progress at the start of the pandemic to develop a product that remains potent and stable in cold chain shipping and storage conditions that are widely available in hospitals, pharmacies, and assisted living and skilled nursing facilities. At the point of care, mRNA 1273 can be deployed in a multi-use vial with no further mixing or dilution, while remaining stable for up to 12 hours at room temperature. Our phase 3 study, which is the basis of our presentation today, was conducted in collaboration with the NIH and in accordance with clear FDA guidance. It enrolled over 30,000 participants, and we believe the results support emergency use authorization. mRNA 1273 efficacy clearly exceeds the recommendations for an EUA and eventual licensure. The vaccine efficacy rate for symptomatic COVID-19 infection was 94.1%, with a 95% confidence interval lower bound of 89.3%. These results are clinically meaningful and highly statistically significant. The efficacy observed is broadly consistent across all evaluated subgroups. Importantly, we also observed a dramatic reduction in severe cases. All of the 30 severe cases observed at the time of primary analysis occurred in people given placebo. A reduction in total symptomatic cases predicts a reduction in cases leading to hospitalization, intensive care, and death. Finally, data from 9 weeks of median exposure in more than 15,000 people vaccinated with mRNA 1273 have well characterized the short-term safety profile. We see generally good tolerability. Most solicited injection site reactions and systemic adverse events were reported as mild to moderate and resolved quickly. It is important to note and to educate people that we see an increased rate and severity of expected systemic symptoms like headache and myalgia after the second dose. We view these as consistent with a potent activation of a specific immune response. They are transient and self-limited, and we do not see a significant safety risk. These results support acceptable benefit risk for broad population vaccination to help prevent COVID-19 infection. We acknowledge the need for longer-term safety and effectiveness data. We will continue to transparently share our data, and the independent DSMB will continue to monitor safety as well as monitoring the duration of immunity and effectiveness. And we will continue to leverage the phase 3 trial, even as we amend it to enable access to participants who receive placebo. Now, in this regard, we face some unique circumstances. First, as it relates to vaccine supplies, none of our trial participants would be quote-unquote jumping the line ahead of others because we have clinical trial supplies available that in fact would expire and go to waste if we don't use them. Second, all of our participants are at increased risk of infection, and many have risk factors for severe disease. One of the participants on our placebo arm died from COVID-19 during this trial. He was a 54-year-old male whose sole risk factor was diabetes. I'll defer to Dr. Baden to describe our proposed next steps on the trial, which will continue to be overseen by the DSMB and should provide significant additional data on both safety and effectiveness. Now, beyond the phase 3 trial, Moderna will conduct additional studies and active pharmacovigilance to gain a more comprehensive understanding of the vaccine risk profile over time. We are initiating pediatric clinical trials, collaborating with the National Cancer Institute to evaluate vaccine safety and immunogenicity in people with cancer, and will continue to collaborate with FDA and other agencies to gather additional long-term safety data. Here now is the agenda for the rest of our presentation. Let me now turn it over to Dr. Melissa Moore.